All right, I see the room filling up, so I'm going to go ahead and get started. Hello, and thank you for joining us. Director of Business Development, Dan O'Connor, will introduce our speakers for today, and Senior Principal Scientist Jim Mullen will moderate our live Q&A. A special focus will be given to the advantages and future opportunities of the PCAT model to describe the processes a drug undergoes following inhalation. We take your privacy rights seriously. By attending this event or participating in the Q&A session, you are allowing us to contact you for follow-up. You may also ask questions via the questions panel on your dashboard at any time. If you need assistance, please use the hand raise icon. Take it away, Dan.
Good morning, all. This is Dan O'Connor, Director of Business Development with Simulations Plus. Thank you for joining us on a great talk today from the GastroPlus user group, the title of which is PBPK Modeling of Pulmonary Drug Absorption, Challenges and Perspective. We're graced with 3 presenters today, a professor and 2 PhD candidates. I'm going to turn my computer off, or my camera off, so that I can read the almost unpronounceable names from the University of Belgrade. So I've already begged forgiveness for mispronouncing their names, and I'm sure that they won't mind terribly. But first, I'd like to introduce Jelisava Ignatovic, who's a University of Belgrade graduate and a PhD student working at the same university, the University of Belgrade. Her PhD is focused on the formulation and characterization of modified dry release powders for inhalation. And she's authored quite a number of papers, both international and national journals, and a number of presentations at scientific meetings worldwide. Additionally, we're joined by Amar Alacevic, who graduated from the University of Sarajevo and has been working on a PhD with the University of Tuzla, completing doctoral thesis in the field of biopharmaceutical characterization of drug products for inhalation. So you can see that there's a common theme here, pulmonary and inhalation. And our first speaker this morning is Professor Sandra Veach, who holds a PhD in medical science and pharmacy, specializing in pharmaceutical technology. She's authored and co-authored more than 150 publications, and her research interests have to do primarily with biopharmaceutical characterization of drug substances, modeling and simulation, disposition and absorption, and bioperformance dissolution testing, and a whole host of other things that I'm sure she'll mention here momentarily. So without further ado, Professor Veach.
Okay, thank you, Dan. I will just shortly turn my camera on to welcome you all to this webinar. And as Dan kindly introduced, today I will give a presentation together with the 2 PhD students, Jelisaveta and Amar. And firstly, I have to stress that our team from the University of Belgrade has been using PBPK modeling for oral dosage forms for years now, but when it comes to modeling of pulmonary drug delivery, we'll still consider ourselves as pioneers. So we are really excited to share our experience today with you. I will now switch off the camera for better connection and keep on going with the presentation. So this is a brief outline of our presentation. The introduction will be quite short because we really want to focus on the case studies and demonstrate some specific utilities and also some limitations of PBPK modeling of orally inhaled drug products. As we all know, GastroPlus software has expanded its modeling capabilities from oral drug delivery to various additional drug dosing routes, and this includes pulmonary drug delivery. This is a picture from one of our recent review papers, and you can see here the list of dosage forms whose bioperformance can be modeled using this specific PCAT model. Yet, literature reports on the use of PCAT models are rather scarce. I've listed here some research papers dealing with PCAT modeling, some review papers here, and also some research papers on PBPK modeling of inhaled drugs, but not using the PCAT model. And although this is not an all-inclusive list, you can surely see that there are not many papers on this subject. And if we are wondering why, we may first consider the challenges of PCAT modeling. This is a representation of the general modeling strategy. And we can see that the first and probably the most demanding step is data collection and the selection of data for model construction. In this step, we can use various experimental or predicted values, but we all know that sometimes it can be difficult to collect all the inputs, and especially to simulate rather complex processes affecting bioperformance of an orally inhaled drug. This is the illustration from one of our papers, which I believe looks quite scary because all of these processes are really hard to simulate. But luckily, PK model offers us a lot of options. Now, I will not describe the PK model in details because there was a webinar on this subject and you can watch it on YouTube. Here's the link. I'll just mention that this module offers the option to input specific compound, formulation, and physiology-related data. And it also contains the integrated models to estimate, you can see here, regional drug deposition in the lungs. And each lung compartment shown here, like, for example, thoracic compartment, is described by a set of parameters which can be modified by the user if needed. So in general, the model has huge capabilities, and you can see here. But it also has certain limitations, and we will address some of them in the following examples. Our first example refers to the PBPK modeling of inhaled amyloid salt. The study plan was to first generate a model describing oral drug delivery, then delivery of a nebulized solution, and finally the model was employed to forecast the performance of 5 model DPI formulations. I've listed some basic biopharmaceutical properties of this drug, and here I'll just highlight that amyloid absorption following inhalation happens in 2 phases. First, quickly via respiratory tract, and then more slowly the swallow portion of the dose is absorbed from the GI tract. So consequently, we have a double peak in the CP time profile, as you can see in this graph. These are the main data from clinical studies. And after careful analysis of the data, we've selected the final data set for the model. Unfortunately, we are lacking time to explain all the inputs, but we have clarified them in our published papers, so you may refer to the reference here. The model was validated for 10 milligram oral solution and 20 milligram oral capsule, but again, due to time limit, I'll just show the data for the oral solution. We've used the whole body PBPK model, and the simulated volume of distribution and elimination half-life matched in vivo value as well, indicating that the model parameters adequately describe the drug's pharmacokinetics. Also, the predicted CP time profile agreed well with in vivo data, which is also illustrated in this table. The model was then adjusted to predict the absorption of a nebulized amyloid solution. The dose was estimated from the declared dose minus the amount of drug entrapped in the nebulizer, which was noted in the reference paper. And the additional input parameters included the selection of dosage form, which was pulmonary solution in this case, and input of mean particle size. And for this purpose, we used mean mass aerodynamic diameter taken from the literature. In addition, it was assumed that the fraction of the inhaled drug from extrathoracic region will be swallowed, And also, in order to match the in vivo data, gastric residence time was prolonged to 1 hour. Now, this is obviously not a physiological value in the fasted state, but it simulates the scenario where it takes some time for the swallowed portion of the drug to reach the GI tract. Here you can see the initial simulation, which provided plausible prediction of the drug absorption, but these values were obtained using the default software calculated absorption rate constant for pulmonary compartments. While some studies suggest that basic drugs like amyloid may exhibit prolonged residence time in the lungs due to lysosomal trapping and therefore somewhat delayed absorption. I need to note here that at the time when we conducted this study, there was no option to include lysosomal trapping in the PCAT model. So in order to assess the influence of decreased lung absorption rate on the generated CP time profiles, we have reduced the initial KAP value by 25%, by 50%, and by 75%. And the insert here shows the enlarged data at the beginning of the absorption process. So, according to the results, the reduction in KAP value had no influence on the extent of drug absorption expressed as AUC, but there were noticeable differences in the predicted Cmax and especially Tmax values. The best match between the in vivo and predicted data was observed with model M3, indicated that inhaled amyloid, similar to other basic drugs, indeed exhibits certain delay in the pulmonary absorption. However, further reduction in KAP value led to the loss of early peak in plasma profile, so model M3 was eventually adapted for further exploration. The model we designed in this study was then used to assess the influence of formulation factors on drug deposition and absorption following the inhalation of 5 model formulations. Aerodynamic properties of these formulations, in particular the emitted dose and particle size distribution, were assessed by 2 approaches, by computational modeling using computational fluid dynamics coupled with discrete phase modeling, and in vitro using Anderson cascade impactor. And you can see from this graph that the predicted values for the emitted dose were similar to the in vitro data, but there were really large differences in particle size distribution, mainly because the agglomerates, you can see here larger particle size detected by the ACI device, could not be considered by the generated CFD-DPM model, which is a huge drawback of the applied computational approach. But on the other hand, ACI results do not necessarily reflect the real situation regarding the presence of agglomerates at the inhaler outlet, because drug particles may also agglomerate and deagglomerate during the ACI measurements. So both methods have certain limitations. Anyway, the predicted regional drug deposition in the lungs based on these 2 data sets and consequently drug absorption profiles, which are not shown here, where you can see quite different. So the question that we want to open here, which will also be addressed in our future examples, is what are the proper inputs to define pulmonary drug deposition in the PCAT model? And having this in mind, we can go to the next example, and I will pass the presentation to Amer.
Hello, everybody. I will talk about our attempt to assess particle size cutoff limits for PCAT modeling. Case study 2 is based around creation of in silico model for beclomethasone dipropionate that was used in process of development clinically and biorelevant dissolution method for preparations for inhalation. First part of that model was relatively uneventful. It is based around clinical study conducted by W. Yates, and it described absorption, metabolism, and distribution and elimination of beclomethasone dipropionate and its active metabolite, beclomethasone 17 monopropionate after intravenous and oral application. Compartmental model was developed using parameters that are presented on the next slide. And as you can see on the presented graphs, obtained in silico results show good correlation with in vitro obtained results. Upper two graph presents results from intravenous application, and lower graph represent the results from oral application. Next slide. These are the parameters that are used in development of model. Next slide, please. Then problems started to arise with design of model for finished medical products. Those are the two pressurized meter dose inhaler products that contain beclomethasone dipropionate. Issue was with incorporation of product-specific parameters in GastroPlus software, because there was no option for straightforward incorporation of results of biopharmaceutical and technological tests that are basis of pharmaceutical characterization of inhaler products. First issue was the easiest to fix, and it was issue of initial dose. Well, catch with inhalers is that the declared dose that you can see on products usually is not the dose that the patient receives. Dose that patient receives is marked as a delivered dose, and procedure for its determination can be found in any pharmacopoeia. And so for start, in the field of initial dose, a value that corresponds to delivered dose was put for each of those two inhaler products. Similar situation was with particle size distribution, and solution was pretty simple. Half of mass median aerodynamic diameter value was inserted in field for mean particle radius, and geometric standard deviation was used for a value of standard deviation. All those values for delivered dose, mass median aerodynamic diameter, and geometric standard deviation were obtained from our aerodynamic assessment of fine particles that was performed on the next generation Impactor, and it was calculated by TITDAS software. But are those two values enough for sufficiently characterized particle deposition in respiratory tract? Next slide. Okay, fortunately, GastroPlus has 3 integrated deposition models, so its users can expand level and quantity of information that can be incorporated in a model. First 2 deposition models are not so forthcoming, and that is understandable, because first one does not have any pharmaceutical background, so to say, and second one does not have much contact with values and methodology that is used for pharmaceutical characterization of inhaler products. That leaves us with the deposition model option 3, user defined, when the user can manually enter percentual values that are deposited in corresponding compartments of respiratory tracts. And when we are talking about values that are used to characterize particle size and behavior of inhaler products in the respiratory tract, we are primarily talking about aerodynamic assessment of fine particles. It is performed on cascade impactor, and results obtained for two tested products are presented at the lower graph. So, but there is no confirmed connection between a result obtained on cascade impactor and deposition in respiratory tracts. And this upper picture that circulates in prospects of manufacturers of cascade impactor for more than a decade does not correspond to a real life situation. Next slide. And that picture is the only existing reference, and it was used as a base for setting out cutoff limits for deposition in a respiratory compartment that are defined in GastroPlus. Anatomical limits for those compartments are grouped partially on the basis of old ICRP66 model, and are represented in a picture, left picture with those red rectangles. Next slide. So on basis of that old picture, basal cutoff limits are set and are marked here as particle size distribution 0. And additional distributions are set by adding or subtracting half a micrometer from those values, so distribution minus 0.5 plus 0.5 and plus 1 are set. And also additional distribution A1 and A2 are set on basis of values that are presented in work by Mitchell and Nagel. And the percentile distribution of particles from 2 inhaler products for each of those defined particle size distribution are calculated on basis of results from NGI by TITDA software. And so simulation are performed on GastroPlus, and the results are presented on this table and graphs. Let's talk about first product 2. The best correlation with in vivo data was obtained with distribution A1, with distribution +1 close by. Concerning data for product 1, issue with that in vivo data is that only one usable concentration time point is obtainable, so correlation cannot be calculated. Instead, the percent prediction error was calculated for that single point, and again, best distribution were +1 and A1, but this time +1 being better. Next slide. So looking at these results and corresponding distribution of aerodynamic particle sizes, we can say that 2 groupings with the smallest errors are +1 and A1. Both of those groupings have particle size limits for alveolar interstitial compartment in similar ranges around 0 to 2 micrometers. Limits for bronchiolar compartments are relatively high compared to other distributions, and they are in a range of 2 to 4 micrometers. Thoracic compartment has lower limit at 4 micrometers, and compartment with higher errors have lower limits set around 3 micrometers. So suitable inputs concerning distribution +1 and A1 have higher values for particle size limits than initially suggested distributions for the in vivo measurement. I'm talking about distribution 0. Modeling results suggest increased particle size deposition in lower pulmonary compartments, alveolar and bronchial, and we conclude that most adequate size distribution in this case are A1 and +1. Next slide. So conclusion. Mass median aerodynamic diameter and geometric standard deviation may not be suitable main inputs for PBPK modeling of particle deposition in the lungs. Percentile distribution of active substance in each of the respiratory compartments could improve quality of results of in silico studies, and groupings A1 and +1 are the most adequate to define the position of beclomethasone dipropionate from PMDI products in this in silico model. Thank you.
I think we now have a time for 4 questions. It was great, right?
Yes, so the poll question is, what modules in GastroPlus are you currently using? And you can select more than one answer here. I'll give everybody a few more seconds here. All right, so it looks like some of you are pulmonary, dermal. There's kind of a good mix amongst everything. That's good to know. Thank you.
Hello, everyone. I will continue now with the 3rd case study. In this study, we attempted to establish the in silico rat model for 3 intratracheally administered formulations using budesonide as a model drug. The first one was commercially available micronized budesonide, or Pulmicort, mixed with coarse lactose as a carrier. The second was budesonide nanocrystal suspension, and the third was budesonide nanocrystals embedded hyaluronic acid microparticles. Budesonide nanocrystals and budesonide microparticles were intended to render faster and slower, respectively, drug dissolution and absorption in the rat lung than Pulmicort. Budesonide is a poorly soluble and highly permeable drug classified as a BCS class 2, meaning that its solubility can be a limiting factor for absorption. The model was firstly constructed for intravenous bolus And then the same drug-related inputs were used to simulate drug performance following intratracheal administration of the 3 tested formulations. The only parameter that was differed was geometric particle size, which was determined in vitro. The results of PK+ analysis show that budesonide pharmacokinetics in rats can be described by 3 compartmental model, and the PK+ estimated pharmacokinetic parameters were used to generate budesonide specific model. 2 types of deposition data were tested as inputs in the PCAT model. First, the values obtained from the in vivo study, marked in this presentation as D1, and in silico predicted values, marked as D2, based on in vitro aerodynamic particle size data presented in this table. Here we just have to note that in the case of budesonide nanocrystals, droplet size was used as input, since this was a liquid formulation, and we could assume the droplet size would be a better predictor of drug deposition in the lungs than the diameter of solid nanocrystal particles. The problem with obtaining D2 deposition data is that the PCAT model does not offer the option to estimate regional drug deposition in rat respiratory tract, so we used ARA software, the multiple path particle dosimetry model for this purpose. Another problem was that the output deposition data from MPPD could not be used as raw inputs in the PCAT model. Namely, MPPD provides data on the deposited drug fractions in 2 pulmonary regions, while PCAT requires data on drug deposition in 4 regions. Extrathoracic drug deposition in this case was neglected because the simulations referred to intratracheal drug administration. MPP data on tracheobronchial deposition were converted to PCAT thoracic and bronchiolar data based on the ratio of surface areas between thoracic and bronchiolar regions. Systemic absorption rate constant from pulmonary compartments was identified as a parameter that needs to be optimized, as I shall explain later. These slides show deposition data for budesonide PT. This figure shows the in vivo data, and these 2 figures predicted values. First, raw MPP data, and then transfer data. The resultant deposition pattern used as input in PCAT model is represented here as budesonide PT redistributed. The same approach was used for all 3 tested formulations. The simulated budesonide plasma concentration time profiles following intratracheal administration of budesonide PT using D1 and D2 deposition inputs indicated 2 peaks in the pharmacokinetic profiles, which are also visible in the mean in vivo observed profile, where the first peak represents budesonide absorption from the lungs, and the second one, drug absorption from the GI tract. The second peak here results from the high fraction of drug dose deposited in the upper parts of the respiratory tract, which was eventually swallowed, and the simulations here successfully captured this phenomenon. As you can see here, both profiles, simulated based on D1 and D2 deposition inputs, provided reasonable prediction of the actual in vivo profile, but D1 deposition input resulted in better prediction of the in vivo determined mean plasma profile, as illustrated by higher R-square value. PCAT modeling also revealed that regional drug absorption patterns from the lungs were similar for D1 and D2 deposition inputs. Now, let's move to the second formulation, budesonide nanocrystal. 2 sets of deposition data are again presented here, and for this formulation, you can notice that the difference between the observed and predicted fractions of drug deposited in alveoli is rather large. When we performed initial simulations, we observed that simulated drug plasma concentration profiles diverged from the in vivo values. A possible explanation was that budesonide in vivo dissolution from the nebulized nanosuspension is faster and more complete than expected based on the initial input solubility value, probably because of the nanosize effect on drug particle dissolution and the presence of additional water in the nebulized droplets. Similar explanation was also found in literature stating that budesonide nanosuspension behaved more like solution than conventional suspension. Based on this assumption, we simulated a scenario where drug solubility and dissolution are not the limiting factors for absorption, so we increased pulmonary drug solubility to 10 milligrams per milliliter, and the predicted plasma concentration time profiles are represented here. And now they matched the in vivo observed profile well. These two plasma concentration time profiles partially overlap, demonstrating that both D1 and D2 deposition inputs resulted in similar prediction outcomes. Although not visible in budesonide plasma profiles, the only differences between outputs based on two different deposition data sets lies in the different pulmonary regional drug absorption. And here's finally the data for the third formulation of budesonide microparticles. The in vivo results for the inhaled microparticles show that the deposited drug fraction in alveoli was again much higher than the in silico estimated value, which can be explained by the fact that current in silico pulmonary models are not able to simulate mucoadhesion of inhaled particles. This particular one was composed of hyaluronic acid as a mucoadhesive polymer. If we look at the CPT profiles, the in vivo data demonstrated delayed budesonide absorption, but the simulated profiles did not capture the in vivo data well. Delayed drug absorption observed in this case can be attributed to the pronounced mucoadhesion of hyaluronic acid microparticles and consequently prolonged drug retention in the lungs. But unfortunately, since the current version of the PCAT model does not offer the option to modify drug pulmonary retention time, nor to use drug dissolution as input, we chose an alternative approach to change drug absorption rate constant. The optimal input absorption rate constant value that resulted in the best matching between the predicted and mean observed pharmacokinetic parameters was this one. The obtained data based on the optimized absorption rate constant value indicate that in vivo determined microparticles deposition in the lungs, or D1 deposition, provided better prediction of budesonide in vivo plasma profile in comparison to the in silico estimated, or D2 deposition. So, in the end, based on the results of this case study, we may conclude that in silico predicted pulmonary drug deposition might serve as a suitable alternative to predict drug absorption following inhalation of relatively simple formulations. In silico modeling may elucidate the difference in drug regional absorption distribution in the lungs, even in cases the differences are not reflected in drug plasma concentration profiles. Prediction of drug absorption pattern following administration of inhaled formulations with pronounced mucoadhesive properties is rather challenging due to the difficulties in determining drug absorption rate constant from the lung compartments. The approach applied in this study, based on the optimization of absorption rate constant, can be used for rough predictions of pulmonary drug absorption, but only when drug plasma exposure data are available to validate the simulation data. However, such an approach is not applicable in the early phases of formulation development. And now I will give a short overview of the whole presentation. To give an overview, highlighted some major benefits and limitations of PCAT modeling. The strength of these models lies in the ability to mechanistically interpret the complex phenomena that happen in vivo and to explore what-if scenarios. These tools are time and cost effective, and they can certainly facilitate formulation of patient-tailored medicines. On the other hand, we are still facing some limitations, such as lack of information on drug biopharmaceutical properties, issues regarding biorelevant input describing drug deposition, and lack of in vivo data for model verification. Furthermore, our knowledge of human physiology is still limited. And finally, we are talking about sophisticated and complex tools, so some level of proficiency is required for their appropriate use. These limitations are actually the roadmap for future improvements. So we hope to see them in the following years. And we also hope to see more research studies on PBPK modeling for inhaled drugs. Well, we came to the end of our presentation, and we appreciate your attention. If you have some questions, we'll be happy to discuss them.
Thank you. So we'll start with the first question here, which is, how can somebody sign up for an evaluation of GastroPlus? And Dan, I think you can answer this one.
Yes, I presume that there's an interest from academia. We have a particular landing page for academics to sign up for evaluation licenses or an academic license. If it's a commercial account, again, if you would want to just send us an email that says, you can send it to me or to anyone that's on this call. We'd be more than happy to establish a trial license for your evaluation. But do they have access to our emails, Jasmine?
Yes, they do. I can put it in the chat, and then they can also go to our website.
Okay, great. Thank you.
I believe we have a few more, Jim.
Yeah, there's a few questions up on the last presentation by Jelisaveta. She mentioned MPPD, and there was a question on how that is calculated. So is there any comment on that?
Yes, thank you for your question. Yes, as I mentioned, MPPD is like another software. So you also need some input parameters. The most important is aerodynamic particle size. And there are also some physiological parameters, but they can be calculated. The software calculates them, so you don't have to input them. So based on these physiological parameters and some parameters such as particle size, the software calculates the deposited fractions in tracheobronchial and alveolar regions.
Alrighty, thank you. The next question is in regards to the first case study from Sandra, so I'll have her answer this one. You mentioned incorporating lysosomal trapping into the model to better predict or estimate plasma concentrations. Are there ways to incorporate lysosomal trapping in order to better estimate lung concentrations?
As I mentioned, because this was the study for the past study, one of our past studies. At the time when we performed simulations, there was not an option in the PCAT model to include lysosomal trapping. So that's the reason why we decreased the absorption rate constant. And yes, it reflected the, its influence reflected on drug concentration both in the lungs and in plasma. But later on, surely when the new version of GastroPlus arrived with the option to include lysosomal trapping, yeah, I've tested the software integrated option to take into account this phenomenon, and the results were quite similar, although the decrease in Cmax and the prolongation in Tmax when I included automatic function of lysosomal trapping was somewhat lower than the one we obtained with 50% reduction in KP value. So it will correspond to about, I think, about 30 to 40% reduction in KP value. And now it is much easier to simulate such phenomena, but at that moment we had to treat the software.
Gotcha. Thank you. Okay, next question. And maybe this would go to Amar, because he talked quite a bit about the impactor data. But this one is regulatory agencies ask for in vivo deposition data for validation of the model. How are we gonna deal with this in the future? Can we address this in the model itself? I mean, you mainly talked about in vitro, but I don't know if you want to talk about deposition in general, Amar, in that question.
Well, my just to say that my opinion is not opinion of a regulatory agency where I work, and my opinion is that in foreseeable future there will be no, I don't see any alternative for cascade impactor data.
Alrighty. Yeah, in vivo deposition is very hard to obtain. So, I mean, what I would like to see in the future is we create a IVIVC going from the cascade impactor type data to in vivo deposition. So I would like us to be able to directly translate that in the future. And I think that was one thing Amar said is difficult to do currently. In the future, that would be what I would like to work on. So there's one question about PD modeling.
Excuse me, Jim, may I add something on the previous question? We have evaluated 3 in vitro methods recently and compared among them about the predictions and also in comparison to the computational CFD model. I think the publication will be accepted soon, so it will be available for the readers. But another problem when thinking about using in vitro inputs as inputs for modeling is that there is a great difference among them. So it's not easy to even establish the correlation between different impactors, and especially between the impactors and the in vivo deposition data, which is, as you mentioned, really hard to obtain, especially in humans. So I think we will face more difficulties when it's about this issue. Unfortunately, I just needed to add this comment.
Yep. Yeah. Have any of you? Sorry, go ahead.
Just a little bit more. When we look at compendial instrument in pharmacopoeias, there is a significant time span between them. For example, Anderson cascade impactor originates from the 60s, from the last century, and next generation impactor is around turn of the centuries, around 95, I think, to 98. And the technology between those two of manufacturing and the limits for particle size cutoffs are getting narrower and narrower. And it's like comparing the Beetle with the new Porsche. You know, You cannot compare those two cars in the same way. Also, that also is valid for the different impactors. Also, impactors are QC instrument for quality control, to make them more usable for correlation with in vivo data. You need to take, for example, the similarity of induction ports. You have that simple USB type induction ports on one side, and on the other side you have idealized induction ports that are more similar to human anatomy, like Alberta idealized probe, or with 3D printing you can print your own scan of your own throat. And also there are also machines that can simulate breathing pattern, different breathing patterns, and that is the way to incorporate cascade impactor data. Not to use the quality control setup that is described in Pharmacopoeia. You need to add spice to that NGI to get data that are more usable for in vitro, in vivo correlation. Thank you.
Alrighty, there's a few completely unrelated questions that maybe I can answer quickly. Is can you predict pharmacodynamic study outcome in GastroPlus? Maybe if you've the panelists have done anything regarding pulmonary in that respect, maybe you can answer. If not, we also provide a pharmacodynamic module in GastroPlus, so you can run models from any of our GastroPlus modules, pulmonary included, and calculate pharmacodynamics in the tissue of action. So that's available. I don't know if anybody has any comment on any pharmacodynamic modeling you've ever done for pulmonary.
And we haven't performed any pharmacodynamic studies regarding pulmonary modeling, but this is a goal within one of our recent projects. So yeah, we intend to do so and to expand our modeling abilities. But for the moment, we don't have any experience in pharmacodynamics.
Gotcha. Yeah, and that was one of the reasons why we put in the additional calculations for lysosomal trapping and calculating better tissue concentrations, was to help link that to our PD module. So that was one of the reasons. There's also another completely unrelated question about, can you model ointments and skin inflammation? And yeah, we have a dermal module. It's beyond the scope of what we're talking here today. So just check our website out there. And I don't see any more pulmonary specific questions. Does anybody have any questions before? We still got a little time, so if anybody has any questions.
Hey, Jen, can you hear me? This is Dan.
Yep.
Just a quick question from my chair. Are there particular data sets that we would be looking to work with any of the folks that are on the call today that might help us expand the pulmonary module? Are there particular areas or things that are in the works where sharing of some data might be helpful to us?
Then if you're asking, yes, we can share the data surely. And if you're asking the audience, I wasn't sure if I hear you right. But yeah, we can share the data that has already been published, and soon we will have more data from the in vivo studies. So yes.
Also, I have more data that should be published in foreseeable future about usage of GastroPlus for clinically relevant dissolution study and building out those models for it.
So we did pick up another question. So this is kind of a pretty broad question. And so how do you understand local and systemic concentrations within the pulmonary module?
Okay, so, may I try to answer that?
Sure.
As as I have a habit to explain to my students, we define the absorption as yes, the amount of drug that passed through the membrane. So if we are talking about the absorption in this way, we may also say that what's in the lungs is in the system. But if we try to differentiate between the bioavailability, something that's available in the systemic circulation and that circulates through the body, and the amount of drug that is concentrated in the local regions, then we can make a difference. But when it comes to terminology, it might be understood in different ways. So if you have something to add, maybe Jim, you may.
Yeah, I mean, it starts with the deposition, right? How much gets into a local area is going to depend on where it deposits in the lung. So that's the first step in predicting the local concentration. The next step is the dissolution, how fast the drug gets into the mucus layer, and then it, as you said, permeates into the cells and the tissue in the lung. And then that's where our model will calculate the tissue concentration. Once the drug's in the tissue, then it's going to then diffuse into the bloodstream, right, and then become systemic. So, obviously we have those 4 or 5 different compartments in the model where we can look at the actual concentrations in the intracellular space or bound to the tissue itself. We have the new calculations in the model that will basically calculate all the drug that partitions into the lipids in the cells, the protein, the lysosomes and whatnot, and it'll calculate the amount bound in the tissue and give you a total tissue concentration. So all those calculations, from deposition, dissolution, permeability into the tissue, partitioning, all those go into calculating the local concentration. And then from there, the diffusion into the bloodstream, the free fraction in the cells determines that. So, that all is going on simultaneously in the model. And yeah, that's what I would say about that question there. Okay, here's another question. Can this model be extended to, or can it be used for extended release or TMDD?
Well, we try to do so with these hyaluronic microparticles with budesonide, but at this moment, it is difficult to model the performance of a modified release product because, in contrast to the model that describes the oral dosage forms, where you can enter dissolution profile, we do not have currently this option in the PCAT model. So we can only change drug solubility to illustrate that it will actually dissolve slower, or maybe change the absorption rate constant. These are, as I think, the only possible options for the moment. So.
Yeah, sounds good. That's been a request for a feature, and that's kind of on the horizon. We plan on adding that feature, so we should have more abilities there in the near future. Then another question we have is, can we incorporate disease physiology like asthma or COPD in the PCAT model?
Yes, definitely, and this is one of the huge benefits of this module. You can change all the physiological data. We just need to have a clue what is going on in this patient population. So if we know that the airflow is somehow changed, or maybe the mucus thickness or something like that, yeah, we can actually test the influence of variations in various physiological parameter values on the predicted outcome. So this is a great opportunity to predict or estimate what might happen in different patient populations.
Thank you. We have another question, again, pretty wide-ranging concentration. So in GastroPlus pulmonary model, how do we optimize the absorption? It says Ka value here. We don't use Ka in in GastroPlus, really, we have permeabilities, more mechanistic description of absorption. Is it manually optimized or software optimized to fit the concentration time curve? Can it be obtained for experiments? What is the last part? Is it possible to have a general model for many kinds of drugs in the pulmonary model in the future? So that's the question.
Okay, a lot of small questions in a big one.
Yes.
There is an option to automatically by the software optimize the absorption rate constant in pulmonary compartments. When you click optimization and then pulmonary physiology, there is an option to select the absorption rate constant, and then the software can optimize them. But in our experience, it was easier to manually test the influence of the decrease in Ka value and the increase, because we just needed to find out what will happen if they're increased or decreased. But if you want to have a very specific value, then you can choose the automatic optimization. There is that option. And as for the experimental determination of the absorption rate constant, we don't have experience with this type of experiments. I think it can be estimated or calculated from the in vivo data. If you have the CP time profiles, you can do a simple pharmacokinetic calculations and estimate the absorption rate constant. But this is not something that we performed. And as for the models with for multi-drugs, each PBPK model is drug specific, so it depends upon the compound properties, and there is not a universal pulmonary model for different drug substances. But you can, for example, generate a file with different records for different drugs, but the model won't be the same. The compound specific data are compound specific. So I think that I covered all the questions, the mini questions.
Yeah, I think that's good. Last question. And this one you kind of already answered with the different asthma or COPD models. And, if you have a variable you want to change in the model, you can. So the last question was, can you mimic different flow rates in GastroPlus? And I think the answer is yes. I'll just answer that one because you pretty much already answered that. So I think that's the last question we have.
Jim, this is Dan. Just let me add, if I may, there was a question earlier about trial license or trying it. If you already have a GastroPlus license and wish to trial any of the modules, we can do that as well. So it doesn't, for those that already have GastroPlus, would like to be trying any of the modules, also just give us a call or a ping, and we'll be happy to set that up for you as well as anyone that would like the complete trial for GastroPlus. Jasmine?
Yes, I just wanted to say thank you to all our presenters. Thank you to everybody in our audience. We invite them to learn more about GastroPlus on our website. That's www.simulations-plus.com. You can also find some of our team members at this year's ACUP conference next month. This webinar has been recorded for playback and will be available on our website and YouTube channel. This concludes our webinar for today. Thank you, everybody.
Thank you, bye.